Here we go. So again, good morning and welcome to the Pacific Internship Programs for Exploring Science, our 2020 PIPES Symposium. We really appreciate all of you taking the time to be here to support the program and especially to support the interns here today. Before we get started, aloha and good morning. My name is Linnea Hugh. I'm one of the program coordinators for PIPES. And although you'll largely be hearing from me, this is a great team effort. So before we get started, I'd like to introduce our team. Sharon, did you want to go first?
Yeah, can you hear me?
Yeah.
Can you hear me? Yeah, cool. Aloha, everybody. This is really weird. We're not seeing each other and we're not sharing our breath, which I guess is the whole idea. But thank you. I'm Sharon Ziegler-Chong. I've been involved with the program since its beginning. So this is our 27th program year of running these kinds of things. And every year I get the same kind of excitement. And this year has been a bizarre year, but it has been really great. You guys, all the interns have given me a lot of energy. So Cherie, you want to go? Pass it to you.
Aloha guys, my name is Cherie Kauʻi. I'm the other program coordinator for PIPES. I'm really excited to hear everybody's presentations and you got this guys. Noi, you want to go next?
Good morning everyone, I'm Noilani Puniwai. I am part of the PIPES Hui here at UH Hilo, but I work at UH Kūpono in the Hawaiian Studies Department. Good morning. Should I tap Lisa?
Good morning, everybody. I'm Lisa Canali. I am the writing coach with the PIPES program. This is my 6th summer, and I'm very excited to see the visual that goes along with the words that I've been reading all summer. And I'm going to hand this off to Becky.
Aloha, I'm Becky Ostertag. I'm part of UH Hilo and part of the Hui of PIPES and been involved in various aspects of the program over the years, and it's always great to see it come to fruition and to hear what the interns have done and how they pull it all together. So that's what I'm most excited about for today, and pass it off back to Linnea.
Sorry. Thanks, Becky. And then also, this is our current team, but we'd also like to acknowledge the previous program coordinators, SAT support people, logistical support people who have been a part of the PIPES team over the years and whose successes we've had the privilege to build upon. Next, I would like to, I would like to express our gratitude to our partners, especially Kamehameha Schools and Hauʻoli Maoloa Foundation.
Bob Peck.
Joined the meeting. To say that this is a different year would be a huge understatement. And we thank our partners for their understanding that operating in this time of a global pandemic could not and would not look like previous years. And so they have allowed us the flexibility to adapt in the ways that we've needed to still deliver this program this summer, and we're really grateful for that. And then last but certainly not least, our mentors and our interns for the summer who, despite the changes and the challenges of the program and creating a remote program in order to care for our participants and our mentors' health and safety, had still said, yeah, let's do it. We're willing to come on this journey with us and learn with us through the process. So mahalo to our mentors from across Hawaii who are willing to take on this new challenge of mentoring remotely and supporting our interns, and of course our interns who are all here today to support and celebrate, for rolling with the punches and being so enthusiastic and bringing the excitement that really drives us forward. So before we get started, this is not our full schedule for today, but it's our first two sort of blocks of presentations. Our final presentation will end at 11:45 today, but we are inviting folks to stick around on Zoom, hang out, and mingle, get to know our interns and our staff in a sort of informal lunch setting. So please keep that in mind. Some really quick sort of housekeeping etiquette things. Again, this is being, for those of you who just jumped on, this symposium is being recorded so that we can make it available to folks who couldn't be with us today. While you are in the session, I think it goes without saying, but please make sure that your mic is muted. Additionally, we're going to ask, because there are a lot of us on here, that at least during the presentations, if you're not the presenter, that you keep your camera off as well. These presentations, single presentations, are 7 minutes long, with only a minute for questions. We understand that it's not a lot of time. And then paired presentations are 12 minutes long with 2 minutes for questions. The timing is tight here, and we understand that, which is why we have 15-minute breaks, as well as informal lunch at the end. So if there are additional questions, which I'm sure there will be, you folks can stick around and talk stories with our interns. However, if you're only able to pop on for a little bit, but still have questions for interns, please be sure to put them in the chat. That will be our way of asking questions after the session, but also if we have too many questions for an intern, then we will log those, and we can email you answers, hopefully get answers to your questions. Let's see, and then, yep, I think that is everything. We have a couple minutes here, so I'm gonna ask if there are any quick questions from folks in the audience, or Sharon, if there's anything that I missed that you wanna be sure that everyone knows.
I think we're all good, Linnea. I just wanted to say to everybody, I really mahalo Linnea for everything she has done this summer. She's really carried this. Sheree and I have been working on lots of things, and she's carried this, and thank you. So that's what I want everybody to know. And let's go. Carried this, and thank you. So that's what I want everybody to know. And let's go.
Great. So first up is Mackenzie Fugett from Kauaʻi. And again, if we can turn off our cameras if we're not presenting.
Okay. All right, aloha everyone. My name is Mackenzie Fugett. My mentors were Uba Nagendra and Katie Champlin, and this is my presentation on the Limahuli Lower Preserve projects and restoration. So just some background information on Limahuli Garden and Preserve. Limahuli is a part of an ancient Ahupua'a system that runs from Mauka to Makai. It's one of the most biodiverse valleys on Kauaʻi, and is home to hundreds of native plants and birds. To keep the valley maintained, there is lots of projects constantly going on around Limuhuli Lower Preserve, but one that I took part in was this rodent control and monitoring project in the Lower Preserve. Introduced rodents feed on a vast variety of fruits, seeds, vegetation, small birds, and eggs. In addition, they also interrupt pollination, nutrient pathways, and seed predation. All of these things lead to ecosystem disruption, and in some cases may even lead to forest collapse. And more than anything, the main goal of this project is to protect our native plant species, protect our native forest birds and seabirds, and protect human health. Our ability to maintain resilient forest areas directly impact downstream communities. If you look in the bottom right corner, you'll see a photo of Cold Pond. This is kind of a local swimming hole for people on the north shore of Kauai. It is near the bottom of Limuhuli Stream, and it's where tons of people gather and play constantly. So keeping everything clean from rat-borne diseases in the upper preserve directly affects this area. This project was started in January 2020 and mainly consists of maintaining an already existing grid of Good Nature A24 traps and Track-A tracking tunnels. Tracking tunnels are this rectangular 14-inch long tube that have an ink card with a dollop of peanut butter placed inside of them. Here is a photo of the actual footprints on the cards. We score rodent percent occupancy basically just on either the presence or absence of these footprints. And then we have our Good Nature traps. These traps were set up shortly before I arrived in June, and what I did was help monitor and maintain these traps. In terms of results, rodent occupancy rates did not significantly change between January and July. In January, there is a 66.67% rodent presence seen on the footprints. And in July, there is a 65% rodent presence seen on the footprints. In terms of traps, between 9 to 36 rodents were controlled. Again, these results aren't super significant, but this project is simply just to lay the groundwork for future ongoing rodent control, and percent occupancy rates should continue to drop as time goes on. So although I got to dip my hands into several projects this summer at Limuhuli, stream restoration was by far my favorite. Stream health is so vital to everything within the valley and beyond, because the stream is what makes Limuhuli a part of the watershed or ʻāhupua'a. Stream restoration is more than just the stream itself. It's the native or non-native forest canopy surrounding the stream. It's what's growing along the banks, and it's even what's going up above in the forest with my rodent control. So restoration takes place in all of these areas constantly all the time, but a big part of this is the stream surveys and monitoring. There are 4 main types that we are looking at when we're surveying. First, we have our O'opu. There's 5 types. We have our Alamo'o, Akupa, Nakea, Nopili, and Naniha. I've seen 4 out of 5 of these, all of which, but Naniha, again, this is still like really, really great odds. It just shows that our stream is happy and healthy. Next, we have our 3 mollusks. We have our hihivai, hapavai, and pipivai. Again, I got to see the hihivai a handful of times this summer, watching them kind of just move up the stream and be happy. And if you look at this photo, this is a photo of the hihivai eggs. So as we're surveying, if you're looking around and you see these little eggs on the rocks, it usually is an indicator that the hihivai are near. The white little capsules are ones that have been recently laid, and the pink ones are ones that have kind of been there for a while. Next, we have our 2 opai, which is our native crustaceans. We have our opai kuahivi and our opai ula. And then lastly, we have our Tahitian prawn. All of these are just indicators of a healthy stream and continue to show us what further restoration efforts we can take. Lastly, I got to work with so many wonderful native plants this summer, but I felt it was important to highlight Palapulai and Kokio. I used both of these plants in restoration efforts, both in the forest and in the stream. And recently I kind of got to dip into the cultural aspect of using these plants by making this lei po'o that's currently on my head. By bringing these two plants together, I'm kind of bringing everything that I learned at Limahuli and even talked about in this presentation with the rodent project and the stream restoration. By weaving these two things together and making this crown on my head, it's really just supposed to show like a physical manifestation of all the knowledge that I've learned this summer. So I just want to say mahalo, mahalo, mahalo. Thank you so, so much to everyone who was involved this summer. Thank you to PIPES and all of its funders. You made this opportunity possible for me. I'm so grateful to have experienced life in the conservation field. And I'm even more grateful that I got matched with Limahuli. Limahuli and all of its staff have been so incredibly helpful this summer and have given me knowledge that I'll carry with me for the rest of my life. And lastly, I want to thank just Limahuli Valley itself. In moments of quiet, it's taught me so many important lessons, and they are memories I will cherish forever. So thank you. My contact information is at the top, and if we have any questions, I will gladly take them.
Any questions from anybody? I don't mean to steal the thunder here. Thank you, Mackenzie. That was such a great, I really, really appreciated that presentation. I've been in Limahuli in the stream, and it's a beautiful stream. How abundant are actually the hi'iwai, hapawai, and pipiwai? How abundant are they? Are they all present all the way up the stream, or what did you see when you were there?
I mainly just saw the hi'iwai. When we started looking in the beginning of the summer in June, we kind of had a hard time seeing them, but as the summer months went on, they really started moving up, and we saw some recently even as big as like the double size of a quarter, which was really, really exciting.
Cool. Oh, that is really, really cool. Great. And thank you. It's such a beautiful valley, and your lei po'o is gorgeous.
Thank you so much.
I think we have time for one more question. If anyone has a question, anyone else has a question? All right, if not, and you think of something in a little bit, you can connect with Mackenzie at one of the breaks or at our lunch at the end of the symposium. I think we have time for one more question. If anyone has a question, anyone else has a question? All right, if not, and you think of something in a little bit, you can connect with Mackenzie at one of the breaks or at our lunch at the end of the symposium.
Thank you guys so much for listening.
So next up, we have Kauʻi Navares. Kauʻi, are you ready?
There we go. Can you hear me okay? Okay, cool. Hello, my name is Kauʻi Navares, and I'll be presenting on the allometric relationship of ridge-top strawberry guava communities. This was a pilot study done with my mentor, Leah Portner, and the University of Hawaii Manoa Department of Geography and Environment. So why is this important? Well, strawberry guava is an invasive plant that has taken over our native forests, transforming the landscape. We were partnered with the Board of Water Supply. They were interested to see how invasive plants affected groundwater recharge. They wanted to know if strawberry guava impacted our water supply. Throughout this presentation, I'll be using these keywords. Allometry, or biological scaling, is looking for the relationship between structure and shape. Hydrology, the water cycle, the movement of water within a system, and transpiration, leaves releasing water vapors. So why did we do this? We wanted to know how water moved within strawberry guava. The significance of this is to be able to scale up measurements to the whole plant by determining the relationship between plant morphological features connected to water movement within the plant. By doing so, we're able to take measurements to determine a relationship, which allows us to take a complex measurement from a simple measurement. These are the measurements that we took. We looked at height, measured from the top to the bottom of the tree, looking at the distance of water being transported, leaf area, the sum of each area of the individual leaves where transpiration occurs, and diameter at breast height, the forestry standard of measuring the diameter of a trunk at 4.5 feet. Our objectives were to look for the relationship between diameter at breast height, and leaf area in strawberry guava, and to produce a technical report with a standardized, detailed methods and suggestions for future studies. We chose 4 field sites along the Liber Ko'olau mountain range. We have Kaunala Trail in Kahuku, Manana Ridge and Aiea Loop in central Oʻahu in Aiea, and Wa'ahila Ridge in Honolulu. We chose these sites because they all share the same elevation, mean annual precipitation, and mean annual temperatures. For our methods, we traveled throughout the Liber Ko'olau mountain range and took destructive samplings of strawberry guava. Since strawberry guava is an invasive plant, we're able to cut down and gather samples. Our field collections included diameter at breast height, and harvested leaves. For a lab analysis, we're able to find leaf area. This is our field site distribution. We collected 72 trees, 18 from each site. These 3 graphs check our experimental design and the differences between the field sites, comparing leaf area, diameter, and height. The ranges are overlapping and are overall consistent. The sites were similar, so we can find the relationship between strawberry guava and not the environment. This is our allometry. We use a linear regression. This data informs us of our field observations. From this, we're able to scale up diameter at breast height and height to predict leaf area of a tree. But we found that a linear regression does not represent the data fully. There was some variations within the data that are not related to the site, but could be due to topography, slope, or canopy status. We took a look at canopy status, and there appeared to be a difference between the canopy and subcanopy. The subcanopy appeared to have a higher height, diameter, and total leaf area that could influence our data. To summarize the results, we saw that the range across sites were overall consistent. Exploratory analysis informed us of our field observations, and the canopy status appeared to influence the different morphological features. This shows us that our research design helped with expanding the knowledge of strawberry guava across Oʻahu. So the importance, again, was to scale up point water movement in a plant, to quantify transpiration, to evaluate the impact of plant invasion on the water cycle. So what did we do? We built a data set that can be continued, created a methodology for the expansion of characterizing strawberry guava stands, and compared leaf area methodologies. So throughout this study, I learned not to be so scared to use R coding. We use that a lot within the project to find our, to do our statistical analysis. While we were in the field, we noticed that the strawberry guava, they all kind of had this fungal infection within them. And within this project, I was also the one that did all the leaf area. And I found that I don't enjoy counting individual leaves to determine the area of them. All in all, it was very nice to have volunteers come and help us with cutting down trees and helping me count leaves. I want to say mahalo, especially to my mentor and all our volunteers that have come with us into the field, and to all of our sponsors that helped fund this program. Thank you for listening to my presentation. Is there any questions?
Mahalo, Kawai, that was wonderful. Do you have a question in the chat? It says, what was the fungal infection? Should we remove strawberry guava from our forest?
The fungal infection that we saw, we didn't really characterize it or know what it was, but something I didn't mention in my presentation is that we also took our sapwood area. So we cut a core from each strawberry guava that we took, and when we looked inside, there was like this black staining in it. We don't know exactly which fungal infection it was, but it was kind of constant through every site that we went to.
Great, mahalo, and Linnea, you can step in anytime. Here's another question. Did it seem like there was a significant relationship between water movement and any of the measures you took?
Okay.
Or a combination of measures?
We saw differences, but we don't know if they're statistically significant. We weren't able to fully dive into like the exact relationship to see the allometry. It was kind of just a pilot study to see if there was something. I don't know if that made sense.
Yep, that's great. And his comment, this is from Cheyenne Perry, he said, I did allometry, I just killed it. I did allometry for gorse. I think I'd prefer vi-vi, ha, ha, ha. Vi-vi, excuse me, not vi-vi.
Oh, okay. No, yeah, currently, a lot of the papers we were reading, there was allometry only looking at biomass of strawberry guava, and we wanted to look at like the water movement and transpiration measurements. So that's why we kind of measured a different, the diameter, base height, and leaf area.
Excellent.
Any other questions?
All right, if you guys have any other questions, we'll let you save it for the break or lunchtime. Mahalo Kawai, that was great.
Excellent.
Any other questions?
All right, if you guys have any other questions, we'll let you save it for the break or lunchtime. Mahalo Kawai, that was great. Next up, we have, so we've gone from Kauaʻi with Mackenzie to Oʻahu with Kawai, and next up Dylan Moniz and Wichimai are going to bring us back here to Hilo.
Okay, sorry about that. Okay, aloha, my name is Wichimai.
I'm Dylan Moniz.
And this summer we worked with Mauna Kea Watershed Alliance, and our mentor was Cheyenne Perry. Our project is on Natural Resource Data Solutions Manual and outplanting preparation. So this summer we worked with Mauna Kea Watershed Alliance. I'm just gonna say MICWA to save some time. They started in 2011, which is also when our mentor Cheyenne Perry was hired as a project coordinator. They're part of the Watershed Partnership Hawai'i Association of Watershed Partnerships, which includes 10 partners from across the state. MICWA's focuses are watershed restoration, forest restoration, native species protection, invasive species management, and outreach and education. Between the Waipahoehoe Management Unit and the Kanakaleo Bird Corridor alone, they manage around 1,625 acres. Those are the two units that Dylan and I got to work in this summer. Our objectives at MICWA this summer were to kokua with the outplanting preparation in the Waipahoehoe unit and to create a user's manual for NRDS to accelerate data collection and analysis. We will be discussing this more in the coming slides.
Okay, so I'm gonna go over Waipahoehoe, which is the main unit we got to work in this summer. So Waipahoehoe is located on the southeastern slope of Mauna Kea. It's within Department of Hawaiian Homelands, and it's located closest to Hakalau Refuge. The elevation inside the unit varies between 5,000 and 6,000 feet. It's kind of steep inside the unit. And so MICWA has been taking care of this area. It's an enclosed, gonna be ungulate-free environment, hoping to restore native species with outplanting and ungulate removal. Some of the ungulates that are within are feral cattle, sheep, pigs, some cats, feral dogs, and various plants. And yeah, well, you can take the next slide.
Okay, so NRDS stands for Natural Resource Data Solutions. This summer, me and Dylan took the time to learn about the app and to create a manual so that the field crew has an accessible on-the-go manual that's easy to understand for whenever they're using this software. So there are tutorials online on how to complete different tasks, but they take time to understand and are not accessible in the field should someone need help to complete a work task. So implementing NERDS in the field data collection will help the MICWA team to take on all the kuleana that they have. It will be a useful tool that can help to accelerate their data analysis process, show whether or not the management techniques that they're using are successful, and it can determine what types of changes need to be made to meet the management goals that they have. NERDS can be used for tasks such as documenting fencing, ungulate control, points of interest, outplanting, and mapping.
Okay, so this is some pictures of the mobile app. So this is on the left side, you can see this is the map screen, which is the home screen on the app. It's basically like a Google Earth satellite imagery. The next picture to the right of that is one of all the different features you can use within the app: tracing, tracking, creating points and polygons. To the right of that is the features tab on the bottom of the app. This is kind of like your glossary or index of things you've created, from fences to polygons to traps. And lastly, on the right side of the screen, if you're on the home screen of the app, it shows the different types of views you can have, from satellite to topography, things like that.
So here's a preview of the NERDS web application. To your left is the dashboard, which shows statistics on like how many miles of fencing they worked on or how many plants they planted. There's also an interactive map, which shows a layer of things like the units that they worked in, as well as fence lines. For example, I use the search bar on the map to find the Kanakaleonui bird corridor, which can be seen on the right. So these pictures are two snippets from the manual that we worked on. On the left is a web application overview, and on the right is a mobile app overview. The steps that we created are very easy to follow, and we try to minimize them as much as possible without compromising the important parts. So the manual will be able to help not only team members of MICWA, but also interns that are coming in and need to learn more about the foundation of the app. I know for Dylan and myself, it took us a little while to familiarize ourselves with how to use it because there are a lot of options that you can use.
Okay. I'm going to talk about the Waipohoihoi outplanting preparation that we are a part of. So MICWA works along an organization that was federally created called NRCS, Natural Resource Conservation Service. And they provide funding to organizations like MICWA. And in an agreement, there are planting practices that are followed. And so on the picture on the left, you can see the area that we worked in. So we would line up tapes along the ground and measure using the spacing practices that is provided by NRCS. And we would spray dots, orange dots every 12 feet. This was basically to create spacing for the plants that will be outplanted in the future. Some of the obstacles that are in the area are the invasive species, the weather, the location, basically the accessibility of the place, resources and how to get them there, and poachers. One of the main obstacles are actually the feral cattle. Not only are they the largest obstacle up there, but they are the hardest to take care of. They are the descendants of George Vancouver's cattle that he gave to King Kamehameha, and MICWA is working alongside Paniolo to remove them by rounding them up and taking them down to be given to DHHL beneficiaries. Yeah. And on this slide, we have some images of the work we did. So in the top left, you can see the plots that we had planted, koa seeds for the future outplanting in Waipohoihoi. To the right of that, the next two pictures on the top and the two on the bottom left are all images of how NRCS gives practices to follow. So there's tapes that go across the unit in each different plot, and there's hiker's tape marked every 12 feet. And what we would do is measure evenly. It is kind of like an orchard style, and spray dots every 12 feet for equal growth between all the trees. And on the bottom right is a picture of ule that is in the greenhouse that MICWA had planted.
So these are some images I experienced this summer. The top left picture is of a pu'u called Kalepeamoa. So Cheyenne actually took us on a tour of the mauna while we were interning with them, and he told us several mo'olelo, as well as different inoa pu'u like this one. On that huaka'i, we also got to see ahina blossoming. So it's always exciting for me because you only see the same plant bloom once in its lifetime. So this image here is of that the flowers. In our time at MICWA, we also spent some time in the greenhouse, as Dylan mentioned previously. So the bottom left corner is a picture of a koa seed that is freshly germinated, and you can also see them a few days later on the top middle picture. So that's the one that was in his photos before that hadn't germinated yet. Finally, the photo on the bottom right is taken from Kanakaleonui near the cabins. That day we got to help with fire prevention by weed whacking the grass, which acts as fuel for fire. It was our only day in the KBC unit, but it was super fun and we got a lot done, and it felt really rewarding to be able to smash it out.
Okay, so these are some images from my experience this summer. On the top left, there's a picture of a stream that was running through Waipohoihoi. You can see all the invasive species, how they're trampling inside the stream and kind of creating erosion on the banks. To the right of that, we have a picture of a hina that we got to see on the Huaka'i with Cheyenne. It's just a very beautiful plant. To the right of that, I just kind of took pictures this summer of these plants that caught my eye, and I'm not sure what this fern is, but it's really beautiful and it caught my eye. To the right of that one is a picture of a koa that I got to see at KBC while working there at the bird corridor. There's just this huge curly koa that was at the bottom. On the bottom left, there's a picture of the feral cattle that's up in Waipohoihoi and in DHHL. These are the cattle that were given to King Kamehameha I from George Vancouver, and they are the descendants of those cattle, and they're very, very intimidating. To the right of that is a Ionui fern. It was the first time I saw this fern up there, and it's just a really beautiful plant. I took a picture, and on the bottom right is, like Vi said, was a day we went to KBC to work. I was lucky enough to walk the fence line with another intern to check for holes and cuts, which we did find from poachers. And yeah, so we basically walked from the top of the bird corridor down to the bottom. It was a good long day of work, so yeah.
So we'd like to mahalo all of our sponsors this summer, and also mahalo Cheyenne and Auntie Nehu, who coordinated everything for us at Mi'kwa, and also mahalo to Linnea folks who worked so hard to support all of us in this endeavor. So any questions? We'll gladly take them now.
As we wait.
Hello, everyone. So, yeah.
Okay, go ahead, Sharon.
Oh, go ahead. No, go ahead, Lynette.
I have one question for you folks. Because you got to take part in so many different things with Mi'kwa, what, as a whole, what do you think that what from this summer do you think you will take with you into whatever's next, be it a job or another internship? What are you taking with you?
I guess I can speak on that. For me, I think flexibility and conservation is something to take away, because before getting into it, I kind of thought of how conservation was just working outside, planting plants, and that's it. But working with Mi'kwa showed that it's a whole bunch of different tasks at hand to play into one big role of restoring the native ecosystem. And so I think it's just being flexible and knowledgeable about all these different things and taking them wherever you go next. It's pretty important. So mahalo for that.
To answer that, from my personal opinion, one thing that I took away from this is that it can be kind of hard to like work with a whole bunch of different organizations who are all managing the same ʻāina. Like there definitely needs to be cohesion, I think, and that's pretty much in anything that we do. But I think it would help to better manage our mauna if there was more cohesion amongst the different organizations that are up there.
Mahalo. You have a question. You have two things from Cheyenne. So, well, one a commented question. One is that you all get milkshakes. That must be an inside thing. Did you folks see any rapid ohia death in the area, ceratocystis?
Yes, as Dylan showed in one of the pictures, there was a bull tied up to the tree. And one of the things that the invasive species there, particularly the ungulates, do is they scar our ohia trees. And so that's basically like an outlet for the rapid ohia deaths to take place. And so we did see a few trees that had already died where we were spraying our dots and stuff, and even as we walked through the forest. And so removing the ungulates, like the horned ungulates, cattle and also sheep would be super important.
Yeah,
Great.
Yeah,
go ahead. Go ahead, Dylan. Sorry. No?
Yeah, I agree with what I said. It's just like she said, the cattle, the bulls create vectors for rod to spread, and yeah, there's not a whole bunch of rod that you can automatically see up there, but there are certain trees that you can tell have gone to it. So yeah.
Great. Thank you. So there's a second question from Maui. Do you think NRDS is a good platform to organize data?
Through our experience, I've actually found it to be pretty useful. And the way that it works is like you can do things on your mobile app without being online. And then as soon as you can hook up to the web or hook up to Wi-Fi again, you can upload it to the web application. And then in that web application, you can see all of the data that you inputted. So it shows you like where your fencing was done. It can track where you hiked that day, so you know exactly where you went if you need to go back there again. And it can organize like all the points that you've collected into a big data sheet that you can later use for analysis.
I think NRDS is super useful. I've actually used an app like it called Onyx, and they're similar apps, and I find it very helpful when you can use your phone and a GPS and input information when you're off grid and nowhere near service, and I think that's really helpful. And I think NRDS is a lot more sophisticated in a sense and has a lot more conservation related availabilities for work from. So yeah, I think it's a very helpful app.
Fantastic. So can you, and this question is to both of you as well, can you talk a little bit, how is the changing climate impacting all of these things that you were looking at? What do you see, and what were the discussions that you had like with Shai and other people? What does the future look like, and how does that shift what they're doing? Or does it?
Whether climate changing, you said, Sharon? Sorry.
That's correct, yeah.
Okay, so yeah, so as we know, like as the climate gets warmer, it also rises up on the Mauna, and a problem that we have is mosquitoes also rising higher up in elevation. And so the purpose of creating the KBC and eventually the second bird corridor is, as temperatures become warmer and mosquitoes rise, the native birds have like pathways to climb higher up in elevation to get away from mosquitoes to help save the population. And I think just as climate's warm, then Mauna Kea kind of, it becomes more affected by, you know, its many ecosystems change, and it's just a vulnerable place. So I think it's important to just continue pounding work over there.
I think Dylan pretty much nailed it. He said what I was going to say. I was going to talk about the bird corridor, particularly Kanaka Nui bird corridor, because like he said, temperatures increase, the mosquitoes can move higher and become more of a threat to our native bird species. So it's important for us to create a habitat for them.
Beautiful. Thank you so much, guys. So we're going to go on to the next one. I appreciate everything. Yeah,
I was going to talk about the bird corridor, particularly Kanaka Nui bird corridor, because like he said, temperatures increase, the mosquitoes can move higher and become more of a threat to our native bird species. So it's important for us to create a habitat for them.
Beautiful. Thank you so much, guys. So we're going to go on to the next one. I appreciate everything. You have lots of comments in the thing saying, woo-hoo.
All right, so taking us from 6,000 feet elevation back down to sea level in the Panaewa Forest are Kūpono Aguiar and Ciarra-Lynn.
Hi, everyone. Good morning again. Thank you, Linnea, for that introduction. As Linnea said, my name is Ciarra-Lynn Parinas. I'm from Hilo on the island of Hawai'i, and I graduated from Hilo last spring. And I'll have my partner introduce himself.
Aloha everyone, my name is Kūpono. I'm originally from Maui, but I came to Hawaii to attend Hilo, where I'm currently in my last year pursuing a degree in environmental studies.
So today we will be discussing developing outreach material for the project that we worked on, which is Liʻikona Pilina. Liʻikona Pilina loosely translates into budding new relationships or growing new partnerships. And the reason why this name was chosen for this experiment was because the team was focusing on creating novel communities within degraded and invaded Hawaiian lowland wet forests, which brings us to where this project is actually located. Keaukaha Military Reservation is a Hawaiian lowland wet forest area. So Hawaiian lowland wet forests were once very abundant in native Hawaiian landscapes, and they had biodiversity that was rich and found in those areas. But however, unfortunately, through time, they become degraded and invaded and heavily influenced by invasive species. So with restoration in mind, the study site at KMR, this area has a long-standing relationship with disturbances, and it consisted heavily of invasive species, which goes into the mission, which I'll let Kūpono explain.
Right, so as Ciarra said, this is all about building new relationships. So what that means for Liʻikona Pilina is that the experimental design is actually a hybrid ecosystem design, and that actually comprises of native and non-native, non-invasive species. The point of this is to try to build ecological resilience and to support native plant regeneration that otherwise could not occur in these highly invaded ecosystems. Yeah, so basically our expectations before going into the internship were that we'd be doing a lot of field work, a lot of hands-on stuff in the lab with our team and other crew members. But unfortunately, due to the COVID circumstances, we were not able to do as much as we wanted, and it changed the schedule a little bit. So our expectations didn't necessarily align with what we did, but we'll talk about that in the next slide.
Very similar to Kūpono, I had my own set of expectations going into this internship. And personally for me, I assumed for whatever reason that we would just be doing like this really rigid and strict reading of academic literature that would lead to this very intense research-based paper product. But as Kūpono said, with the way that our internship unfurled with all of the restrictions and limitations, the dynamic became very like fluid and flexible. And we'll get a little bit more into that.
Yeah, so a lot of the stuff that we actually got to do was we did get to go into the field. So what we kind of did was half in the field, half remote work. When we were in the field, we got to get lots of hands-on experience with different kinds of native plants and culturally significant non-native plants. Right here is my hand picture with some noni fruit, and I personally love noni juice. So getting to see all these plants in person and take different measurements, data collection. We did census work where we tracked the growth. We used height poles, all kinds of really cool stuff in the field, including data collection. One of the samples that we did have to collect were the arthropod samples that my partner will talk about for the microscope section.
So if you notice, there is a bottom right portion that's a leaf litter collection tray. So leaf litter was actually collected a few times when we were out on field, but for the arthropod sorting, after we collected that litter, we actually set up release funnels with some of our senior members, preserving them in antifreeze. And after we were assigned samples, we actually, we looked at our samples while we were at home. So on the top row, you'll see both Kūpono and my setup from home with our microscopes, just like on our tables in the middle of our house, but it was so much fun. Essentially, what we were doing, we were looking at the samples that we were given, which we've also provided a picture of in the top row of what our samples look like underneath the microscope. But the ultimate reason why we're sorting and looking through these arthropods is because arthropods are essentially the next trophic level that this experiment can observe and run tests on. Each plot was designed with plant function, with tree functions in mind. So by collecting and eventually later identifying the arthropods, we can gather data to test any significant, or if there are any significant associations between plant functionality and arthropod functionality, and we can begin making those assumptions of community development.
Yeah, so based off of our field work and some of the lab stuff that we did, we came up with products that we felt were necessary to increase exposure about the project. Prior to us joining the project, there was a project website that wasn't necessarily the most up-to-date, which is understandable being that websites are really difficult to update frequently. So I decided to make an Instagram, partly because of the photo sharing capabilities, but also because it's easy to update with day-to-day activities. Some of the tags I created helped people organize information and basically like pick what you wanted to learn about the project. One of my favorite ones was the Likonapilina species profile. And what that is basically highlights of all of the native species and the outplant species in our plots. It talks about things like where they're native to, what is their cultural importance, and even their ecological role in the project. For me, what the Instagram really was about was helping people build relationships with these plants, because I felt like the more relationship you had with something and the more you understood it, the more you would be inclined to protecting it. So even for people that aren't necessarily conservation minded, I tried to include things like medicinal uses, food uses, fiber uses for other people that may not be in conservation, but still have connections to these plants and understand their importance. I also got to do a little bit of data analysis. And one of the things that I did was a piercing correlation between daily average minimum temperature and percentage of individuals fruiting each month. I found somewhat of a correlation between the two, but it wasn't exactly linear. So my thinking is that there's some kind of temperature threshold that needs to be met before the onset of fruiting occurs. And the reason why I chose fruiting is because the presence of fruits implies the presence of seeds. And seedling recruitment is really important in forest regeneration and building ecological resilience overall.
So luckily, with how social media is, there is an option to scaffold Kūpono's material from Instagram to Facebook. So with that, I really wanted to give the Facebook component, which was my component of the social media development, a different lens or a different view. So where Kūpono was focusing on sort of the information, I really wanted to actually focus on humanizing the project. That was the main purpose of this organizational Facebook account. When we think of Facebook, we think of friends and family. But the fact of the matter is that it's a crucial tool for information and influence potentially. So instead of just having the data and statistics on our account, the focus would show the people behind the project and what they've done and how they have gotten the project to where it is today. And that would be by using a hashtag Throwback Thursday with old pictures of gathered and archived from the project to kind of complement what is going on today or what we've gathered for our internship. Facebook also is an opportunity where we can actively interact with the community instead of just sort of like passively sitting and just watching or just liking or commenting. We'll actually have the opportunity to like share like potential DIY projects or recipes that use the species from the experiment with the community. The live streams may be an option, but in all honesty, live streaming from the field does pose some sort of logistic difficulties, and it also depends on the approval of KMR, since there are some things with our social media development that has to be approved beforehand. But last but not least, it's a quantifiable way to kind of gauge your engagement with the community, which is great data to go into the future. While the Facebook was my sort of smaller project, my main project was actually a game. I'm a huge fan of games for education because they serve as an alternative method for information, mostly because my personal belief has always been that information should be accessible and applicable and relevant for everyone. Learning should be fun, which is why I decided to sort of reboot a card game. So essentially, my outreach product is a card game based on a Hanafuda card game specifically played in Hawaii, which is Sakura. That's the specific game that we play, and the cards are there for your reference. But essentially what I did was, instead of the traditional Japanese designs, I replaced the 12 associated plants with the species of our project. And it's simply a matching game where you aim to acquire the most points. And so while there are small differences between the instructions which have been laid out, the important part of this game is actually the variant supplementary lessons that come along with it. These are just outlines of potential discussion points for players to sort of think about and ponder on after they play a quick game, like species interactions and invasive species. But the important thing to me was to include varying difficulty levels, I guess you could say, for like different students who can come in contact with these topics, mostly so that way we can sort of introduce and approach like these big conservational restoration ideas at a younger age with more digestible content.
Yeah, so overall, I had a really great experience. I did eventually get to do field work, which was great. Getting experience doing data collection, samples, and all the hard on-the-ground stuff really allowed us to get a lot of background on the project, and I feel like we developed really good outreach materials because of it. I just really learned the importance of getting in the field experience, because it's difficult to make, you know, outreach, Instagram, stuff like that if you're not really there and you don't know what's going on. So that was a really big part for me, was that I got to see two sides of the coin of this whole conservation project.
And for me, this was my first actual experience with conservation, professionally and academically. And it was definitely eye-opening. For a short 10, I know 10 weeks doesn't sound short, but we all know that it is short. And in that time, I have reached so many profound conclusions, or so many profound, like, oh, this is where I want to start my research. Let's go from here on forward. Because just knowing the sheer manpower and the insight and the communication and everything that needs to go into a successful conservation project and restoration project, it's just such a beautiful thing to be a part of. And I'm very grateful for this opportunity.
Yeah, and of course, we like to mahalo everyone at PIPES for being a great support to us, not only through our projects, but with everything else we were working on at the time. And of course, the crew at Likonapilina for taking us in and really like accepting us into their family, teaching us so much. I personally learned a lot. They helped me with my photos, a lot of really cool ideas. So yeah. And I think we're good. So yeah, if anyone has questions, I think we have some time for that.
Have a lot of applause going on in the chat here, guys. And one of the things is, what's the Instagram handle, whatever, I don't know what you call it. What do you call it?
Well, I guess we have so that's the Facebook, just Likonapilina. You can search that up. And then the Instagram is also Likonapilina. We don't have any content yet because it is a military reservation. We do have to get all of our posts pre-approved. So we're sending out our draft content for approval. So, but we will be posting soon, hopefully.
That's great. So lots of applause. Any questions for these guys? Let's take a question. And in not hearing one, I'm going to ask you the same question too. Again, what does this look like with adaptation to climate change? There's so many elements that you were looking at here, both with people and with plants, right? What does that look like and going into the future as things shift? Things are always shifting, but what does this look like?
Oh.
How will it impact this project?
I think it's really cool that you bring that up, because on this slide right here, we actually have photos of the different canopies in our treatments. So on the right side, you can see that the ulu leaves do a really good job of suppressing the weeds and like preventing invasion. So that was really cool to me, the way that this project uses non-native species to support the native species and to increase seedling recruitment of natives. So it really goes to show that like traditional methods of conservation that are like native only are great sometimes, but in some instances, we do have to think outside of the box. And these hybrid ecosystems were my first, I guess, taste of what that looks like. So yeah, moving forward, I think opening your mind up to stuff like this, that could work.
That's great. Ciarra, if you have something to add, and then as long as I have the mic, for Kūpono, what's next in your fruiting study? Will it continue?
I'm hoping to explore other aspects because we didn't look at like rainfall and even like soil composition and stuff that all affect fruiting. Maybe even looking at other aspects of phenology too, because we also do have flowering data. So I don't know. I just have all new questions after doing my data analysis.
Okay, I think that's all the time we have. I'll pass it back over to Linnell.
All right, thank you, folks. All right, thank you, folks. We are a few minutes over time, but that's okay because we are on break. We will be back from this break, and our next presentation will start promptly at 11:20. So please be sure to be back in your seats to catch it maybe a minute before it starts. But for now, it's a good time. Sorry, thanks, Shri. 10:20, not 11:20. I'm getting my times mixed up. We'll be back at 10:20 for our next presentation. And so, again, this is a good time to stretch, use the bathroom, do what you gotta do. And we look forward to seeing you folks back here in just a few minutes. Aloha and welcome back, folks. We have about a minute until our next block of presentation begins. If you are just joining us, a few housekeeping rules. We do ask that as you come in, you make sure that your video is off and that you're on mute so that our presenters can continue uninterrupted. If you have questions, please feel free to put them in the Zoom chat, and one of our staff will be helping to facilitate questions to our interns after their presentations. We have a short amount of time, about 1 minute for questions after presentations, and so if you have additional questions that you would like to ask of our interns, you can leave them in the chat and we'll get them to our interns and get you answers. Or you can stick around or come back to join us at lunch, which will begin at around noon today. It's just an informal opportunity for us to sit around and talk story and ask questions. All right. Well, buckle up, folks, because this next block is really exciting, especially for those of you who love Hawaiian birds. We actually have 4 projects this summer. That's a third. That's almost a third of our cohort. But 4 projects that focus on native Hawaiian bird conservation and research. And research. And to kick off this block, we have Kaitlyn Evans from Maui.
All right, aloha everybody and mahalo for being here. My name is Kaitlyn Evans and I had the privilege of working with the Maui Forest Bird Recovery Project during this PIPES internship term. Today, I'm just going to share with you a little bit about my journey that I have been on during this summer. So because this internship was a little different given the current circumstances, I was tasked with a wide variety of mini projects by the MFBRP team that I could work on from home and be remote. So my main interest is actually in environmental outreach and education. So when I was tasked with social media planning, that was a real treat for me. I got to help the MFBRP team plan out some of their Instagram posts, and I was able to bring kind of that refreshing outlook on the work that they do and be able to communicate that to the public. I also did a lot of bird blood sample and banding data entry, and I also helped them edit their East Maui plant guide. And then when I finally got to make it out into the field, we got to do a lot of seed collection out in Kula Forest Reserve. And I also got to camp out in the Nakula Natural Area Reserve, which is actually the main project site location for the big project that I was working on throughout this entire summer. Over there, we did some erosion control by installing some coirs, which you can see in this top picture. And we also did a lot of native outplanting. You can see from these pictures also the intense erosion that's out in the Natural Area Reserve. It was really shocking to just walk by some plants and see their roots completely exposed, and that just showed how fast the soil drained off in such a short amount of time. Along with all of those mini projects, I also got to visit and help out a bunch of other organizations that help to contribute to helping these native birds, including the Olinda River Plant Facility, the Maui Bird Conservation Center, and Nu'u Refuge. At the Olinda River Plant Facility, I was able to help care for the nursery plants, and I got to see how these plants were kept before they were picked up by other organizations for various restoration work. And then at MBCC, I got to see the work that went into keeping these native birds happy and healthy. And I also helped to assist in prepping some of the birds' meals, which you can see in this picture over here. There's like a bunch of bowls laid out. It was pretty crazy to prep all of those meals. And then at Nu'u Refuge, that was really awesome because we got to spend a lot of time digging into the actual history of the area and tying all of those cultural aspects and the archaeology of that area together. So for my big project, throughout the entire summer, I helped MFBRP create an updated report that featured their avian point count survey data that they collected from 2015 to 2020. And a point count is basically taking various observations that, like whether that be through hearing or seeing the different birds and being able to detect them in that area, and then you basically count off the amount of detections that you have, and then they compiled all of that. So the report helps to analyze the bird composition out in the Nakula Natural Area Reserve using this point count data, and it helps us to observe the current bird distribution there. They are also currently restoring the Wailaulau unit in the Natural Area Reserve. So this report can also help to analyze how the bird population responds to the restoration as that habitat starts to shift. So in the future, the current restoration will help to aid in creating a more suitable habitat for these threatened native bird species, since this is one of the last places that these birds are actually able to survive out in the wild. As we got further into analyzing the data, though, we found that there were a lot of variabilities, and that could alter the actual trends that we ended up finding. So future studies will need to account for that. So this is an example of the different kinds of trends that we were able to discover when we were analyzing the data. So this map shows the average number of apapane that were detected throughout the Nakula Natural Area Reserve. Each dot on the map represents a point count station along each of the 3 different transects. So we can see here that there's a higher density of apapane in the upper section of the NAR and little to none at the bottom. So from these types of maps, we can start to make inferences to determine why these trends are actually occurring. So for the apapane, their trend could be due to the species having a preference for the upper elevations, or even a preference for that specific habitat located up there if we start to analyze the map using the actual satellite imagery. But we still have no or too little data to make any real conclusions. This can, however, be a tool in the future when more data is collected to help show the progress of habitat restoration in the NAR and how these individual species start to respond to it. And it can also help to determine the each species' niches. So this PIPES experience has been a real journey for me, and I'm very glad I got to be a part of this cohort despite COVID-19 making so many modifications. This period forced me to adapt and roll with the punches. And although I applied to PIPES hoping for a lot of field work, I was happy that I got to delve into the office work and learn how to appreciate it. Working remotely also helped me to better my communication skills and make sure I was constantly updating everyone on the status of all of my mini projects and asking for questions and help whenever I needed it. This experience gave me the opportunity to learn all the hard work and the actual process of writing a scientific report with lots of guidance. And I know that this will greatly benefit my future in this field. Being able to work with all of these different aspects that go into the work MFBRP actually does reminded me of the importance of community and a team effort. I learned that not many locals go into work with native birds just because we don't have the same access to study them as the continental US does. So being able to engage the community by having programs that will help to educate and expose locals to this work is very crucial, especially in helping to connect people to their place and getting them to see why caring for their place actually matters. So mahalo piha to all the organizations that made this journey possible, to PIPES and the PIPES cohort for your support, to the MFBRP staff for your guidance and your knowledge. Mahalo nui, and I will now take questions if you guys have any.
Mahalo, Kaitlyn, great job. Do we have any questions for her? So I have a question, Kaitlyn. What were some of the significant uses or importance of these birds back in time?
I would say a lot of their main purposes was like actual native seed distribution, and that's kind of how restoration ties into the importance and like the cycle of keeping these native birds surviving and thriving, because planting these native plants will help to, you know, create more seeds for them to feed on, and then therefore they're able to then spread those seeds elsewhere and facilitate the restoration. So it's a very cyclical process and reciprocal process.
Awesome, thank you. Do we have any other questions for Kaitlyn? Okay, Sharon has one. How do we increase the participation of local next-gen scientists in bird recovery and restoration?
I would say a big way, well, nowadays is social media, just because so many young people are on social media. So that's kind of why I'm wanting to go into outreach and education, is because I want to continue to expose younger people to the work that conservation does, and also locals seeing other locals in these fields, because it makes it more, you know, like local people can relate to other local people. So it makes it less, I guess, intimidating in a way. And it motivates other locals to that, because they're able to see that there's other like people like them doing this kind of work. So yeah, definitely outreach, education, social media, and being local in this field. So yay to all the locals that are here.
That's a really great point, Kaitlyn. Okay, so I think we'll turn it over back to Linnea to introduce our next presentation. Mahalo, Kaitlyn.
Mahalo.
Mahalo, Kaitlyn. That was an awesome presentation. Next up, we're gonna take it to Timon Skinner, who is going to tell us about one very special Native Hawaiian bird.
All right. Aloha, everyone. My name is Timon Skinner, and I work in the LOHE lab, which is the Listening Observatory for Hawaiian Ecosystems. We use bioacoustic science to monitor the health and activity within ecosystems, especially those of Hawaiian forest birds. My project for this summer was to study the vocalizations of the Akiapola owl, and my mentors were Brett Mossman and Pat Hart. So to start us off, let's get some background on the Akiapola owl. It's a endemic Hawaiian honey creeper. It's endemic to Hawaii Island. There are incredibly limited population numbers. There are less than 1,000 alive today, and they are they're threatened by the same things that threaten other native Hawaiian birds, like predation from rats, mongoose, cats, habitat loss, and especially mosquito-borne disease. But they're really unique in their foraging methods, which they've adapted a bill that is suited for sort of a woodpecker niche, which means that they drill into the wood of koa trees and use that elongated upper beak to dig for invertebrates and like larvae. And due to their rare and elusive nature, they are relatively unstudied. The project that I'm doing is sort of the first of its kind. No one's ever done a formal study on Akiapola owl vocalizations. They live high up in the canopy. They forage exclusively on koa, which means that, you know, as the forests disappear, our ability to study them disappears. And they also don't like to vocalize very much. So you have to kind of be there at just the right time. Luckily, we have the Hakalau Forest National Wildlife Refuge, which is sort of like a sanctuary for a lot of native Hawaiian birds and also endangered plant species. The elevation provides a good buffer from mosquitoes. It's at really high elevation at about 5,000 to 6,500 feet. And reforestation efforts have created this young koa canopy that's only about 30 years old, and that has helped sustain the Akiapola owl populations. And the lack of undergrowth actually makes it a lot easier to follow them around, because if it was a true koa forest that was highly developed, there would be a lot of brush to work your way through. And luckily we can still study these birds while it's relatively easy to get to them. And so in order to study these birds, we use bioacoustic science, which basically involves going out with a field recorder and pointing it at these birds. And luckily, Akiapola owl can be identified by the range they inhabit, since they're super territorial and they don't really venture far into overlapping territories. Luckily, my mentor had already observed the territory range of several of these birds prior to my internship. So we were able to just go straight into their range and follow them with handheld recorders. And when we made the recordings, we also marked the GPS location of the tree that we observed the Aki to be in. So further down the line, we can potentially visualize the territory of these individuals and the mating pairs. Now, my project specifically focused on the differences between individual vocal repertoires, or in other words, comparing the vocabulary of individual Aki. Now, to do this, I took recordings of 3 Akiapola'au and prepared them for analysis in the software called Lucinia. And what you see in that box is sort of a time lapse of the process. So the green lines, they outline each element of a syllable, which is basically like, it translated, that's the notes of a song. Each element is a note and each syllable is a song. And the red square dictates the length of each song. And these markings set the parameters that are used for analysis, which is the frequency range and the change over time. Now, once all the vocalizations are prepared, they can be analyzed within the Lucinia software. And I use this method called simple comparison, which basically compares the contents of each song to every other song. And personally, I didn't really know what to expect, but I found that there appears to be some grouping of syllable usage. We had a really small sample size. There were only 3 Aki, but there were enough vocalizations to create this similarity matrix, which, to break it down a little bit, each of these is, oh, shoot, my bad. Each of these is an individual vocalization, and however close they are to each other shows how similar they are. And so I noticed that as there were more songs for each bird, there appeared to be more grouping. This top grouping right here was, we called him Aki 5, and he had the least number of songs. He only had about 20 songs. And this middle grouping was Aki 3, and we had about 40 songs for this bird. And then all the way down here is Aki 1, which had the biggest cluster and also the most songs, with about 80. And as you can see, there are also a lot of similarities. I think, horizontally, that means that they're in the same frequency range. So the notes might not be exactly the same if you're comparing from all the way over here to all the way over here, but they are within the same range of frequency. And in conclusion, or the conclusions that we came to, are that Akiapola'au, they occupy their unique frequency range, and they appear to share some notes, but they may sort of remix them. And there might be some viability to identifying individual Akiapola'au by their vocalizations, but there needs to be a much larger data set to actually back up these claims. And what's next after this? Ultimately, I want to collect things of individual Akiapola'au, and once we've done that, we kind of will have addressed one of the main downfalls of this project. Oh, sorry, I'm kind of over time, huh? Yeah, I'll try to wrap it up fast. So once we can collect more of these recordings, we can also compare the birds by location. And ultimately, I'd like to comprehensively document the Aki vocalizations and use them to sort of inform conservation efforts of the forest that they live in. And last but not least, big mahalo to my mentors, Brett and Pat. They supplied an incredible wealth of knowledge and resources, and I was able to camp up at Hakalau during COVID, which was such a blessing. And also mahalo to everyone at PIPES, Linnea, Shuri, Lisa, Sharon, Becky, and Noe. I can't believe that this even happened, so I feel really lucky. And thank you for your support and helping to navigate through these turbulent waters. Oh, sorry. And any questions?
Awesome. Mahalo, Timon. Great presentation. For folks who may have questions for him, please stick around during the break and or at our informal lunch. But awesome. Mahalo, Timon. Great presentation. For folks who may have questions for him, please stick around during the break and or at our informal lunch. But we're going to turn it over to our next speaker, Russ Masuda.
Hello, everyone. My name is Russ Masuda, and this is my presentation, which is on proving the accuracy of neural network for the detection of bird songs. Okay, so over the summer, I worked with the Listening Observatory for Hawaiian Ecosystems, or aloha, with Patrick Hart and Grady Weinberg to improve the PEPL program. The PEPL program uses neural networks to automate the time-consuming process of identifying bird calls within audio data. This increases the processing rate, allowing for more data to become available. However, some issues with this program is that it's difficult to use and returns a high number of false positives. So over the summer, I was tasked with improving the precision and recall of the existing PEPL program and creating a graphical user interface for it to make it easier to use. Okay, so what are neural networks? Neural networks are deep learning algorithms that perform classification. They start by trying to convert an image, say an image of a cow, into a numerical array. After that, that numerical array is passed into the neural network, and it assigns weights and biases to the areas within that array itself. Then it undergoes a process of resizing the array and updating its weights before setting input a label. Okay, so as I said before, to pass the audio file into the network, it needs to first be converted into an array. So in order to do so, I used a short Fourier transform. In order to improve the network, I believe that normalizing the data between 0 and 1 could improve the performance since the segmented function was used at the output layer. So the very first thing I did was I performed the absolute value transformation on the data itself, thus changing the data from 1 minus amplitude. Then after that, I performed a log 10 transformation to make the data less skewed to help to assist with the learning of the new network, followed by a min-max normalization. Okay, so as before, the goal of this project was to improve the precision and recall of the different neural networks within the PEPL program. Precision is a measure of how many false positives there are inside the predicted results, with values of closer to 1 that indicate fewer positive results. Recall is a measure of how many false negatives there are in the predicted results, and values closer to 1 indicate fewer false negatives. Okay, and here are my results. In the convolution model, I saw a decrease in precision by 32% and an increase in recall by 21%. Within the Grill and Schaefer bubble model, I saw an increase of precision of 23%, with a slight decrease in recall of 2.6%. And within the GRU model, I saw an increase in precision of 17% and a decrease in recall in 6.3%. In addition to normalizing data, I also created my own transfer learning model in the network itself. Idea behind this was for the larger was for the neural network itself to learn basic concepts from the larger ResNet 50 network, and then add additional weights so the network can learn specifically from the PPL audio files. However, this did not work well because the training data was too different from the ImageNet dataset. As such, it was unable to leverage the performance to relation and performance decreased. Okay, and I also was tasked with creating a graphical user interface for the network itself. Originally, users had to type in commands to run the program, as seen towards the right. So I implemented a graphical user interface to make the program itself easier to use. Okay, this consists of a home screen, input screen, future screen, train screen, and predict screen. Here is the home screen and input screen. Okay. And here is the PPL feature screen where data preprocessing takes place. By hitting the Add Files button, a file dialog box opens, allowing users to select whatever files they want to input to use to train the neural network. And by hitting Submit, data processing takes place, and the results are displayed to the GUI as it occurs. Next up is the train screen. Same thing occurs with the Add Files button. And by hitting Submit, you can use your preprocessed files to create the model for the neural network. Finally, within the Predict screen, you can add your testing wave files via feature dialog box. You can select what model that you trained, and then you can output the results to the GUI itself. Okay, in summary, by normalizing the training data, the precision of the GRU and bubble models increased, however, the recall decreased. Using transfer learning methods with the ResNet 50 model with audio data did not work well at all. And I also implemented a graphical user interface for the PPL program. Here are my image references. And I'd like to take the time to acknowledge the University of Hawaii at Hilo, the Waialae Mauna Loa Foundation, Kamehameha Schools, Office of Mauna Kea Management, the Quiet Forest Bird Recovery Project, the Listening Observatory for Hawaii Ecosystems, specifically Christine and Anne. And the software was originally written by the 2018 AKY REU research students. Of those students, I'd specifically like to mention Madore Rampanelli and Lindsay Howards for the work on the project. Thank you very much. Do you have any questions?
That was awesome, Russ. Thank you. We have one question here from Travis Mandel. How does this compare to other neural networks for birdsong identification?
How does this program specifically compare to other neural networks for birdsong identification?
Yes, that's like what the question is.
I'm not really too sure. I'm not like really aware of other neural networks that are used for this type of work.
Russ, would you mind sort of reiterating or returning to how, obviously this neural network, there's still needs to do some learning, so there's some improvement that can happen. But you know, a fully functioning and well-taught PPL program, how do you see that contributing to bird conservation and research?
Personally, I believe by like automating the process of like labeling these audio files, we could actually create like more data that is available that be used for both environmental research and conservation research.
Wonderful, thank you. Oh, one more. We'll take this one quick question. Yoko Uehara is asking, how long did it take to process your data through your deep learning?
Okay, so with this neural network, I believe I processed over 15-minute, I believe I processed over 15-minute wave files in roughly 1 hour. That includes data preprocessing, training the model itself, and predicting the results.
Sharon, thank you so much. That's you are in a world of a language I don't even understand. That's really cool. It's really fun to hear you talk about it because it's such a different world. And so, do you see yourself continuing in this? Is this something that's really exciting?
Yes, I find this type of work very exciting. So yeah, absolutely.
Yeah, cool. Great. Thanks for a great presentation.
Thank you.
Great. Thanks for a great presentation.
Thank you.
All right. So rounding out our bird block, it is Paul Dill-De Sa, who will be presenting about the aloha. Cole?
All right. Thank you for everyone that came today. My name is Cole Dill-De Sa. I worked with the Hawaii Endangered Bird Conservation Program, the HEBCP, and I thought my project was pretty cool. They basically gave me a bunch of data and they said, hey, do something with this. So the question we all settled on was inbreeding's effects on aloha mating behavior. All right, so broadly, lately the status of endangered species has become an increasingly relevant issue to environmental conservation efforts worldwide. This has to do with biodiversity, which plays into general ecosystem health and vitality. But bringing it into Hawaii and the focus of this presentation, it's the aloha, which is the endemic crow species to our islands. Currently, it's facing extinction due to various human-caused interactions with the Hawaiian ecosystem, mostly invasive species and human development in their habitat. And this is important because the aloha actually plays a pretty critical role in the health of our forests. They disperse seeds and help repopulate our native flora. Okay, so focusing in on the project itself, which is to do with inbreeding, conservation efforts are underway to restore the bird's wild population and save the species from extinction. And a part of that is HEBCP has a large number of aloha birds kept between their two facilities, one here in the Big Island in Volcano, KBCC, and one in Maui, MBCC. But because of the nature of their founding population, there are only a couple of birds caught from the wild and a couple of eggs collected. The birds that they have are pretty highly inbred, and the question of whether or not this has an effect on their behavior is something that's not really known, and something that we decided would be worth studying. And so the data that they gave me is almost a decade's worth of observational pair data that the HEBCP researchers have been collecting. Basically, it's studies, or it's just observational notes on how they interact with each other, some statistical stuff like number of eggs, chicks, other things like that. So jumping right into methods, using all of this data, we decided to compile an Excel spreadsheet using this Survey 123 software. And I have a picture of sort of what I was working with. As you can see, we tried to isolate variables that we determined were important to answering the question of behavior affecting, inbreeding affecting behavior. So this has to do with pair compatibility, aggression, destruction of eggs. And here we have, I don't know, you can see some of the other things like nest building, cup forming. Those are all pretty important measurements. But another really important thing that we used was the inbreeding coefficient of each pair. So this is basically a number between 0 and 1 that's assigned to each pair to determine how inbred they are. 1 being inbred, very inbred, basically the same individual, 0 being not related at all. And from previous research by HEBCP, we determined that a value of 0.1 is like used as a threshold marker to determine an inbred from a non-inbred pair. All right, so results. So first figure here is compatibility versus the mean F value, which is the inbreeding coefficient. So a higher value is more inbred. As you can see, actually birds that were more inbred got along better than their counterparts. And I thought that was pretty interesting. Figure 2 is the proportional likeliness to damage or eat eggs split between inbred and non-inbred pairs. So this is where the 0.1 F value comes into play. Above 0.1, so of the pairs that damaged or broke their eggs, there's a it's far more likely for inbred pairs to have exhibited this behavior versus non-inbred pairs. All right, continuing on. Figure 4 is sort of the same idea of the last figure, splitting it between inbred and non-inbred. It's a proportional likeliness to display aggression between inbred and non-inbred pairs. And again, inbred pairs were more likely to display aggression towards each other than non-inbred pairs. And figure 5 is, which I thought was pretty interesting and just kind of used to sift through the data a bit more. It's basically the mean F value of aggressive inbred pairs. So it's this bar right here, the inbred bar in figure 4 versus the mean overall F value. So as you can see, aggressive inbred pairs are a bit more inbred than the overall population, which is to be expected. Yeah, so conclusion. From our results, we conclude that within the data set that was given, there does exist a measurable difference in observable behavior between inbred and non-inbred aloha pairs. And specifically, it implies that certain behavioral tendencies, specifically egg damage and aggression, are more likely to occur within inbred pairs versus their non-inbred counterparts. And this information is useful for the HEBC husbandry staff to make more informed decisions regarding pair matchups and basically generally help our, or help inform our knowledge of the bird in general. Yeah, but it's hard to draw an absolutely resolute conclusion because of the nature of the data, right? Much of it was based on observed variables from each researcher. So it adds sort of a layer of subjectivity to the research. And I don't know if you guys noticed, but back in the other figures, the sample sizes of the aggression and the egg damage graphs were between 20 and 30% of the entire population. So more data should be probably collected to make a more resolute conclusion on this topic. But nonetheless, it provides a promising conclusion regarding inbreeding having an effect on aloha mating behavior and could be a stepping stone for more research in the area. Oh. just yeah, I'd like to thank all these people, the people who funded PIPES, KS, Hilo, Hau'oli. And I'd also like to thank my mentors, Allison Flanagan and Russ Masuda. They helped me a lot over this past 10 weeks, and San Diego Zoo Global.
Awesome. Mahalo, Cole. Great presentation, really interesting. Do we have any questions for him at this time?
Hi, this is Paul Banco. That's great presentation, Cole. I'm just wondering if you think that the aggressive and egg-breaking behaviors are reflect just the difference in individuals' response to being in captivity. Not that that would have any bearing on whether they're inbred or not, but maybe the inbreeding index that you come up with is really more of a reflection of their response to captivity, or maybe not. Do you follow what I'm meaning?
Yeah, that is an interesting point. I didn't actually think of that in this project, and it very well could be. Yeah, I just think more research kind of does need to be done to answer these questions. But I don't really know how I would test that, because all the ʻālala birds are in captivity right now, versus not including the ones that have been released recently. But yeah, that's a really interesting question.
Yeah, I agree. I think we may have to wait for a time when there's wild ʻālala whose pedigree we know and their degree of inbreeding, and to see whether their egg breaking and aggressive behavior is similar. And also there might be an age effect that would be worth considering, that maybe older individuals would exhibit less aggression, maybe because they're more habituated to captivity. But anyway, I think those are very interesting results.
Thank you.
Cole, we have a question from Vi. What behaviors do ʻālala exhibit towards other native birds?
Again, it's hard to say, because we don't have a lot of research on wild ʻālala. Everything that has been collected, or everything that I worked with at least, has been from the ʻālala kept in the two facilities split between the Big Island and Maui. So yeah, I think there are a lot of interesting questions that, given time, we can answer regarding these topics.
Okay, and then we have one more question from Natasha. Are there any efforts to continue observations over time?
That I'm not too sure of, but I was actually thinking, as far as inbreeding is concerned and behavioral tendencies, it would be really interesting for HEBCP or anyone to sort of do the same thing that they're doing with the observational data, except sort of define it in a methodological process for inbreeding. So, like I said, a lot of it was a bit subjective, like what exactly is aggression between pairs? Because one person could define aggression in a different way than another. And so making a more comprehensive data set going forward would be really helpful to continuing research like this.
Great. Thanks, Cole. Good job. I will turn it over to Linnea at this point.
Thank you.
Great job, Cole. And way to round out, I'm going to say it one more time because I love it, our bird block. We have one now. We have so many interns doing bird projects that we can group you all together. So that is the end of that block. We are going to take a quick break. Everyone can stretch and have some time to walk around. We will be back and starting our next presentation promptly at 11:15. So please be back before then. And remember, I'll be hanging around here, so if you folks have additional questions, feel free to put them in the chat. All right, see you all at 11:15. Okay, so again, welcome back. Thank you for joining us today. We have one more block of presentations to go. Just a few quick etiquette reminders for those of you who are just joining us. Please be sure as you join that you keep your video and as well as your mic off. Like I've said before, we will be having an informal lunch hour after the presentations are over, at which time we can all turn on our cameras and there'll be opportunity to talk story with our interns. If you have any questions throughout these presentations, please be sure to put them in the chat, and one of us will make sure to ask the intern. If we run out of time for your question, please join us at lunchtime to ask the interns in person, or if you need to jump off the call, we can keep your question and make sure it gets to the intern so that they can get you an answer. All right. Well, I'm going to stop sharing because, so this next block is a collection of internships that are really focused on community, whether that be our human communities, our ecological communities, or sort of the interaction between both. And first up is Jacob Wessling. All yours, Jacob.
Thank you. All right. Aloha mai kākou. My name is Jacob Andrew Wessling, and my project for PIPES this summer was the demography of Hawaiian coral reefs with Dr. John Burns and the Multi-Scale Environmental Graphical Analysis Lab, or for short, MEGA Lab. So first, a little something about me: Hawaiian Marine Science, Conservation, the MEGA Lab, and PIPES. I was raised in Ewa Beach on Oʻahu. I was a 3-minute drive from the place I learned to surf. I was either out fishing or out surfing. I think one thing growing up that I wonder if other local people can relate to is I just hated seeing the negative changes, you know, going back to our favorite spots, seeing the change of corals or the change of coastline. And that was something that I wanted to preserve for future generations. Now onto the project locations. So Papahanaumokuʻākea, or the Northwestern Hawaiian Islands, is 120,000 square miles, stretching from Nohoa Island to the west of Niihau, all the way to the Kure Atoll. This stretch of archipelago represents just about 70% of the shallow water tropical coral reefs under the US flag. With that said, more than 7,000 species call this place home. This includes fishes, invertebrates, sea turtles, and marine mammals. And out of that 7,000, 25% are endemic. So if you look at the map down here, you can tell that Papahanaumokuʻākea is significantly bigger than the main Hawaiian islands we live in. Now onto Lalo, or the French frigate shoals. So Lalo is an open atoll with a large crescent-shaped reef surrounding numerous small sandy islets. If you look at the figure above it over here, you can see that the crescent shape of the reef covers over 938 square kilometers or 232,000 acres. That's pretty huge. With that said, what makes Lalo so cool is its coral growth. And it's atop of this eroded volcano, which has been submerged for millions of years. And it has a steep-sided basalt pinnacle that sticks out in the middle of the water. That's the last remnant of the original volcano. Another reason why Lalo is so cool is it has the greatest coral biodiversity in the Northwestern Hawaiian Islands. So with that said, it has 41 documented coral species. Now onto our project. So what are we looking for? We know that the community of coral types shape survival. So we're trying to look for demography. We're looking for the strongest community that corals thrive within. Is it all table corals, all branching corals? Is it mounding corals? Is it a mix? We don't know. That's what we're trying to find out. I have an example of a tabling coral down here, which is a Copra Cytherea. It's common all throughout Lalo, but essentially it's absent from the main Hawaiian Islands. Now, so To survey these huge reefs, we have to first gather images of the subject. Dr. John Burns created a method that has divers go out to specific coordinate points in Lalo, where the diver uses a DSLR camera in a dive housing, of course, to shoot hundreds of overlapping photos of the reef below by swimming in a back and forth lawn mowing motion. So if you guys follow my cursor, right here on the diver will come swim right across here, taking photos, and it will loop back around and do the same thing. So he or she feels that they have enough to make a 3D orthomosaic. So this method allows the photos to be 70% overlapping, which gives us, the researcher, an easier and clearer picture when making 3D models. So now, when the photos all have been taken, I can then download them all into an Agisoft software called ArcMap. Here, I render all the photos into an orthomosaic, which is a bunch of photos stuck together like the one below, and it gives it a 3D model. So for example, this is about a 20 by 5 meter orthomosaic that contains thousands of coral colonies, all at various sizes. I know it may look small, but this is a bunch of different orthomosaics stuck together, made up of thousands of pictures. Pretty crazy. Now on to digitizing. So the next step that we do is we start digitizing coral species. So once the 3D model is created, I input the orthomosaic into another Agisoft software called ArcGIS. Using ArcGIS, I create circle plots that is randomly put all throughout the model. So if you look above step 1, this is an example of an orthomosaic with randomized circle plots all around the model. And next, when that's finished, I will then trace any coral patch or colony within the circle plot. So this includes coral that are 50% or more showing in the circle plot or has the center of the coral showing also. So right above, you can see I trace different corals in these circle plots. Now, when all the corals have been traced in a circle plot, I can now identify species, morphology, structural complexity, even the length and area of these corals. So all of which is shown in the figure right above step 3. I have some patches of Acropora cytherea that is have been digitized. So this is what it looks like after. And our last step is we record the information. So we like to call ourselves the Census Bureau. You know, we're asking these corals, you know, how are they doing? What species are they? Are they bleaching or not? So that's why we kind of like to call ourselves the Census Bureau, which is kind of funny. Now, how we study corals, people seem to ask why. Why are corals so important? The coral reefs are one of the most important and productive ecosystems on Earth, even more productive than tropical rainforests when it comes to fixing carbon and creating oxygen. Many communities around the world rely on these reefs for not only food, but protection, and many communities here in Hawaii also. With that said, we hope our study can give us the critical information we need to protect and preserve our coral reefs. And like our main goal is just trying to find what attributes and what constitutes a strong and resilient coral so we can make that community for them and we can protect them. So what's next? We are still working on digitizing. That is pretty much the whole bulk of our project. 80% of what we're doing is all digitizing. It may look easy to trace corals, but you guys can come sit down next to me and see how easy it is. But with that said, we're gonna keep working on this study so we can get results. And I just wanna keep learning. Ever since I started learning about reef ecology and corals, I've just been wanting to keep learning more and more. And with that said, just keep going into conservation with Dr. John Burns in the Mega Lab. It's been so much fun and I hope I can keep doing it for as long as I can. Mahalo nui loa. Dr. John Burns, the whole Mega Lab, Kylie, Kilani, Alex, all of you guys, thank you so much. The PIPES program with Linnea, Cherie, Lisa, Shannon, Noi, Becky, thank you, all you guys. And for our funding with Kamehameha Schools and the Ha'oli Maloa Foundation. Thank you.
Awesome job, Jacob. Really cool project. Do we have any questions for him at this time? I like that that Census Bureau comment.
Yeah, I like it too.
So we have one question from Travis. How do you know how many circles to look at?
So our circle plots, we actually get as a template. So it's all around the same amount, which is I think it's under 30. But yeah, we have a template that we put in to our ArcGIS, and it gives us the whole randomized circle plot. If that answers your question. Okay.
I have a question. How many hours did it take you to sit in front of the computer and make all of those dots?
Oh man, I can't tell you how much, actually. I don't think there's a number. It's just, I mean, you just sit down and then it's 9 a.m. Next thing you know, you look and it's 1. Or it's just, it does take a lot. A lot.
We have a question from your mentor. So how was the process of learning all of the coral species?
It was a long but gratifying process. You know, when I first started, it was a 200-page PowerPoint, and it was just like, here's all these corals. Study them. You're gonna be seeing them in underwater, so you just know them. And I was just dumbfounded and about just how many there were. But I think by just breaking it up through families and geniuses, it made it really easier. And it took a couple months. I started like right when schools stopped, and I think now I could say I'm, like what John Burns said, a Coral ID Jeopardy champion.
Classic. Okay, right on, Jacob. Mahalo. That was a great presentation. I'll turn it over to Linnea to introduce our next speaker.
Thank you, Jacob. And thank you, Sarah. I see you have a question, and we'll make sure that that gets to Jacob. Mahalo.
Right when schools stopped, and I think now I could say I'm, like what John Burns said, a Coral ID Jeopardy champion.
Classic. Okay, right on, Jacob. Mahalo. That was a great presentation. I'll turn it over to Linnea to introduce our next speaker.
Thank you, Jacob. And thank you, Sarah. I see you have a question, and we'll make sure that that gets to Jacob. Mahalo. Next up, we have Kauʻi Aguiar, who is joining us from Kona.
Okay. Aloha mai kākou and good afternoon, or excuse me, I guess it's still morning. Good morning. My name is Kauʻi Aguiar. My name is Kauʻi Aguiar, and I had the immense privilege this summer to work with Dr. Marina Carides of the Island and Indigenous Sociology track, and my focus was on the relationship between Kanaka and their natural surroundings and resources, and how the lack of access to those resources has affected Hawaiian culture and subsequently Hawaiian identity. As I dove more into my research, that focus became more specifically tailored to a relationship with the kai and haulawai'a, or fishers. Divers and gatherers reconcile lack of access to their cultural practices and sense of self. In order to explore this, I conducted interviews with local Kanaka Lawai'a, Kia'i Loko, or local i'a caretakers, divers, and gatherers, as well as in-depth analyses of likewise literature. I would like to start off by giving a brief definition of culture and what I mean by effect, and then talk about why I personally chose this topic, show a few examples of what me and my interviewees discussed, and then convey to you my final thoughts. So, how I define culture was shared beliefs, ideologies, customs, and social institutions held by a group of people. So pre-contact Kanaka Maoli cultural practices were very much dictated by the reciprocal relationship the people had with the ʻāina, or land, and the kai, ocean. This relationship fostered the understanding that Kanaka Maoli are related ancestrally to their ʻāina. Post-contact Hawaii, however, brought not only new ideologies and new social institutions with the introduction of Christian understanding and Haole influence, but the notion that indigenous language, customs, and cultural practices were, in a word, no longer necessary. So when I say affect, my meaning is have those influences and now lack of access change the way that we as Hawaiians look at ourselves. I found myself drawn to exploring this topic because as a Kanaka in Hawaii today, I often feel like parts of my understanding of myself and my heritage are missing. The connection that Hawaiians have with their home is crucial, I think, to their sense of self, my sense of self. With the near loss of Hawaiian language, loss of freedom, and loss of jurisdiction over land came a generational deterioration established by colonization of pre-contact culture. Because of the opportunity I've had to explore the relationship between Kanaka Maoli as a whole with their ʻāina and kai, it has also afforded me the chance to explore that personally, and I'm very grateful for that. So I'm going to provide a few quotes that were thematic amongst my interviews that were informed in grounded theory, and that will be explained more in Kauʻi Kalili's presentation after this. And in the interest of confidentiality, I am using pseudonyms or fake names. So in the first quote, I asked 30-year-old Lawai'a Alika what he thought about when he was in the ocean or being able to gather. And I know this man personally, and he is usually somewhat stoic and very straight-faced. But as he opened up about his concerns for the kai, he cried and heartfeltly expressed to me his love and concern for the ocean he had known growing up. He said, "I think about the development, the pollution, and sometimes the lack of fish. Where they at? They used to be here, but why aren't they here anymore? And all the laws they're trying to inflict upon us to make it harder to gather." Second is 32-year-old Mahi'ai and Kia'iloko Mahea, who eloquently conveyed how she needed to be continuously culturally practicing, for it quite literally gives her a reason to live. She said, "For me, ʻāina, vai, and kai, and loko is integral to my well-being. It's integral to the point that I will probably commit suicide if that access was oki, if that was cut. It makes me think a lot about our kupuna and the heartache that they went through when the mahele came, and that eviction and that dispossession and the continual dispossession." And lastly is 52-year-old Keoni, who is pretty much straightforward with how he feels his duty and others' duty is as Hawaiians. "There's a responsibility," he says, "that comes too with being Hawaiian and knowing that the people of Hawaii manage their resources. They took care of it. You don't overfish an area if you want to constantly be going to it. And I think that being Hawaiian, there's hopefully an understanding of that." I feared that the progression of the world of Hawaii brought with it ways of thinking that could have been at detriment to Native cultural understanding. All hope is not lost, however. My grasp of Hawaiian culture, my own culture, was and still is limited. I have spoken to many Kanaka, especially in my generation, who feel the same way. The reason I believe for this is the increasing lack of access to native lands and gathering locations, and regulations that consistently prohibit Kanaka but are favorable towards those who have the means to restrict said access. Moreover, those restrictions do not affect Kanaka in mere superficial ways. As I found out from my interviews, it affects Native people's livelihoods, psychological stability, emotional stability, and especially identity as Kanaka Maoli. What I also found was that a common theme from every person that I spoke with was a deep love and reverence for the kai, and almost all related that back to being Kanaka Maoli. These conversations allowed me to see that there was still many Kanaka who are active cultural practitioners and gatherers, which has given me hope for not just myself, but for our future keeping. I want to give a big mahalo to all of you for listening, and Dr. Marina Carides, Linnea, and all the PIPES folks, and Kauʻi Kalili for being so supportive of me and helping me with my research. It was really a blessing and a great opportunity, and I'm really thankful. Thank you.
Awesome. Mahalo, Kauʻi. Really interesting project and great presentation. Do we have any questions for her at this time? Okay, so I have a question. What are the next steps in this project? Where does this go from here?
I really think it has to do, besides the lack of access, I think there are a lot of people, a lot of Kanaka who advocate obviously for more access, but I think also, especially, and I can't speak for everyone, I guess I can only speak for those I've talked with and myself. I think in younger generations it is crucial, and I heard other interns talking about it, for education. I think there aren't a lot of opportunities besides maybe like familial opportunities or going to Kamehameha schools or explorations. I think there aren't a lot of opportunities for Kanaka now to learn about themselves, you know, if that makes sense. And I think if there were more programs like that, you could perpetuate that culture, if that makes sense.
Cool. Okay, so we have one question from Ku'upua. Did you look into the case law associated with traditional and customary rights for your research?
I did not, although I will look into it now. I actually, yeah, I did not. I don't know what that is. The case, can you repeat that? The case law?
Did you look into the case law associated with traditional and customary rights for your research?
Yeah, I don't know what that means. I'm sorry.
No, that's fine, Kauʻi. This is Sharon. I mean, case law is looking at the established court cases that have focused on these kinds of rights and approaches, etc. So, and I think you did not do that, but that's fine too. Great presentation. I really like this. Thank you to you and to Marina as well. There's another question, Sheree.
Yes, another question is, how would have cultural perspectives changed from pre-contact times to present day?
So that was an aspect that I focused on, which was slightly hard to have like grasping information on, but what I recognized was that because of the lack of access and because of things like fish depletion and obviously new inventions like electronics and things like that, our cultural perspectives on ourselves have changed because our relationship with our ʻāina or with our kai have changed. If that makes any sense.
And then you have one more question. Let's see. Are you planning on continuing your relationships with those you have talked with, potentially for graduate school or additional interviews?
I wasn't sure about continuing for graduate school, but I'm continuing those relationships with those people. I thought that was very important to keep speaking with them, and I've actually been continuously speaking with a lot of them and developed great friendships out of that. So that has been really great for me.
Okay, great. Thank you so much, Kauʻi. I'll turn it over to Linnea to introduce our last speaker of the day.
All right, and Sarah, I just want to let you know that we see your question, and that's a really great question, and that's a big one. And so I think that if you are available to join us for lunch, that would be a great start to our talk story. And if not, we'll make sure that your question gets to Kauʻi and that we get you an answer as we can. So next and last is Kauʻi Kalili.
Aloha, I'm Kauʻi Kalili, and my research project for this summer is Hula, Transferring Environmental Knowledge Across Generations. I had the privilege of working with Dr. Marina Creedis as my mentor and the organization Hilo Sociology Program. My presentation considers hula as a form of environmental science. To evaluate, I interviewed hula dancers at various stages of skill and practice, as well as investigate the literature. I'll begin by defining hula and providing some history and discuss how Native Hawaiians use hula as a vehicle for transferring environmental knowledge across generations. I will then turn to my interviews with hula practitioners to show how they have come to understand hula as a form of environmental science. Here's some history of hula. Hula is a traditional Kanaka Maoli dance form, but there was a time that hula was banned during the colonization of Hawaii by the United States in the 1800s. At this time, Christianity spread rapidly throughout the island, seeing hula to be immoral, causing that ban. Eurocentric ideas of success and influence of Christianity caused generations of Hawaiians to separate from cultural traditions, practices, belief systems, and the language. Many Native Hawaiians at that time were taught that success in life required that you abandon the Hawaiian language and culture. That same thought transcended down many generations. Other perspectives by Noino Silva, who is the author of Political Economy of Hula, argues that the stop of hula was also imposed due to economics. Her examination of this discourse reveals the legal ban of hula was related to the problem of cheap labor needed for sugar plantations. Hawaiians then see hula took focus away from them working on the fields. Fortunately, some Kanaka Maoris in the past were able to hold on to traditions, enabling Kanakas today to carry on those same traditions and see hula as a way of life. Hula contains cultural and genealogical context, but also explains the natural environment through a Native Hawaiian lens. Interactions of environmental forces and environmental conditions of Hawaii can be explained through hula. During my research, I reviewed many literature that relates to the study. One of them is from Adrienne Chapler, who is an anthropologist who studies dance in Oceania. She prefers the term structured movement over dance, as dance is a Western concept, typically and does not typically recognize dance as a form of knowledge or ritual. Chapler recognized that indigenous dance forms are more than just movement. Those movements hold cultural values and deep structures of society. I use perspectives by Dr. Pualani Kanahele of Halau Kekuhi and Kumuhula Peleka'io of Halau Unulau. Kanahele relates experiences of the dancer to that of the environment. She notes that the sweat that is released by the dancer to that of the environment, like transpiration and evaporation of water from the forest into the air. But it is a movement of the sun that controls this process. Kanahele explains that hula feeds the environment by mimicking it. Now I'll speak a little bit of my own personal journey. As a hula dancer in Unulau, hula has a profound impact on me and my environmental awareness. Growing up as a Native Hawaiian woman, I always felt a disconnect to my Hawaiian culture. My Hawaiian grandparents were raised in a time where it was shame to practice ancient Hawaiian culture. Their shamefulness transcended on to my father, who also had that same understanding. My grandparents and father had a lot of knowledge, but never realized that significance of that knowledge. In 2017, I became a student of Halau Unulau under the direction of Peleka'io. Hula has helped me foster a deep relationship with our natural environment. I have been able to view our natural world from a different perspective, that I have never before had, a perspective from our ancestors. My hula practice has shaped who I am as a Hawaiian, who I am as a mother, a sister, a daughter, and a positive contributor to our community. Now I'll return to my data analysis and our interviewing process. For my PIPES research, I conducted seven semi-structured interviews with students of Halau Unulau via Zoom. I interviewed 4 Olapa students who are intermediate level students and 3 Ho'opa'a students who are the advanced level. I transcribe these interviews and I use grounded theory approach to analyze these interviews. Grounded theory approaches qualitative data with the goal of deriving theory from the perspective of participants through the development of codes, themes, and eventually theory, but comparing interview text and codes. Concepts of themes developed that are clearly grounded in data, providing new understanding of this social phenomenon. 4 themes emerge in my data. 2 of them in which I'll discuss today: environmental kinship and Kanaka guiding. Environmental kinship. Hula is first an environmental dance in which the kuahu or shrine in the hula practice space is available to serve dancers with a reminder of that connection. Forest pans are placed on the kuahu as it is a shrine. Environmental kinship is the relationship between people and their surrounding environment. The theme environmental kinship developed from my interview participants discussing how hula practice sharpened their connection to the environment. In the interviews, culture and environmental connections seem to overlap. It seems that commitment to the process of hula is reciprocal. It is the commitment to self, community, family, ancestors, the environment, and society in general. I will use pseudonyms as I share personal quotes by my interview participants in interest of confidentiality. For example, Kanoe states, "I'm committed to this hula lifestyle and being a hula person. I mahalo being in the Unukupukupu Hala'okekuhi genealogy tradition. It helps to ground me in who I am as a Kanaka. The environmental connections to water and land that I have acquired has helped me do my job better in conservation." Lehua says, "My commitment to Unulau has drastically refined the path that I am on, and it has helped me deepen all experiences in my life by understanding how to act and behave and contribute to my community." The importance of Kuahu was a common theme brought up in my data. Manu explains, "My commitment to hula is my commitment to the forest. The Kuahu is my forest." As Kumu Huakanahele explains, the reality that hula is first a dance of nature keeps us alert of the well-being of our heaven, earth, and skies. My next theme is Kanaka guiding. Colonization of the Hawaiian Islands has caused many Native Hawaiians for generations to lose touch of who they are as Hawaiians, causing many to question their culture and beliefs. Many Native Hawaiian practicing hula use hula as an outlet to build strong Kanaka identity. It has become too easy in our world today to lose touch of foundations passed on by our ancestors. Cultural foundations such as hula help Kanakas live in a world today that is constantly evolving. Kanaka guiding has helped Kanakas understand their place in the universe by building environmental connection. For example, Kelani says, "Hula has enabled me to become an environmental steward, where I'm able to expand my own knowledge. Through this process, I have been given opportunity to create discipline in my life. Hula has taught me leadership." Iolani says, "Hula for me is a commitment to my self-growth and my commitment to the forest. I will always be committed to hula as long as I know I continue to contribute to the space because it is contributing to me." Haumea also says, "Hula for me and being in halau has made me more confident in what I know as a Kanaka and what I choose to follow. People can say so many different things and I don't have to follow their truth. Through hula, I have found my own." In conclusion, the themes I've developed from my interviews help to identify how hula is an important vehicle for maintaining and passing along environmental knowledge. However, for contemporary practitioners, it also solidifies Kanaka identities and provides a path of greater kinship with the environment, a kinship to the environment that has helped shape a better quality of life for those hula practitioners, enabling them to be positive members in society. I would like to give a big mahalo to all the awesome PIPE staff, my mentor, Dr. Marina Corrientes, and Halau Unulau for their participation. Also, Kauʻi Aguiar for being so supportive in my research journey. Mahalo, any questions?
Mahalo, Kauʻi. That's a great presentation. Do we have any questions for her at this time? So it looks like we have a question from Natasha. Was the banning of hula in Hawaii gradual, or do you know, or did it happen over time? And if so, In historical context, is it possible to identify periods in Hawaii's history when the practicing of hula was banned and a decline of environmental stewardship has coincided?
I don't quote me on this, but I believe that it was gradual. There was one of our queens, who is Queen Kauʻi, who actual banned it because she was involved in Christianity. But after she moved out of office, we were allowed to dance hula. King Kalakaua was also big on that revival of hula. And I'm sorry, I missed the last question, the last part of that question.
In historical context, is it possible to identify periods in Hawaii's history when the practicing of hula was banned and a decline of environmental stewardship has coincided?
Yes, I didn't dive too much into that, but if you were to do your own research on history, you definitely see the decline. And like I said before, a lot of Native Hawaiian people believe that if you practice your own culture and dance hula, it negatively impacted your life and you weren't as successful in life. So a lot of people moved away from it. And I can say that for my own grandparents, they didn't really care to learn the language because they thought that they were ashamed to do it because they thought they weren't doing what Hawaiians supposed to do. Like the Western way was the best way.
Awesome. So you have a question from Malu. Have you looked at Mele speaking to and or supporting your themes of research?
My plan was to look at Mele, but I didn't get that far. But I plan to continue on my research journey and dive into that. That's a good question, Malu. Thanks.
Awesome. We have any more questions for Kauʻi before we wrap up? Okay, great job, Kauʻi. Mahalo, that was a great presentation. I will turn this over to Linnea to tell us what's next.
Woo, you guys did it. Mahalo, mahalo, mahalo to everyone for being here and to all of our interns for pulling together such wonderful presentations and representing our program and your mentors and your ohana and yourselves so very well today. Actually, I'm gonna stop sharing for a second because this is a time when we could put our videos on. And if you folks are comfortable, do you mind putting your videos on just so the interns can see the faces of all the awesome people who showed up for them today. And just awesome job. Guys, that was wonderful. And thank you. And yeah, there's no such thing as applause really on Zoom, but you can dance, you can shaka, you can smile, whatever it is to thank Sharon for really letting them know what an awesome job they did. This ends the formal part of our PIPES Symposium. Again, just thank you. We know that a lot of folks have Zoom fatigue. We know that your time is precious, so that you know, you choosing to be here with us for a part of your day really means a lot. Sorry if it's windy and you can't hear me as well. So really quick, before we sort of break again, before our informal lunch hour, I want to say a thank you to our team, to our partners, to our mentors and interns, and all of the alumni and supporters who are here, who are you know, supporting from afar and in their hearts. but I also want to say that part of what we do in PIPES, like obviously none of it can be done without the partnerships and relationships that we have been fortunate enough to cultivate over the years. And in the spirit of not just you know, supporting and cheering on our interns, but students from across the university system and other campuses, I wanted to ask Marata Tamaira from the Islands of Opportunity Alliance, Louis Stokes Alliance for Minority Participation, that's here at UH Hilo. They are another program that's doing really great work with students here at UH Hilo, but also across the Pacific. And they have their conference that has gone live. So I'm gonna really quickly share, here we go. Share their slide. And Marata, if you would like to come on for a sec and just plug your folks' awesome student conference that goes live today.
Kia ora koutou, everybody. My name's Marita. Thank you so much, Linnea, for opening up this space for us to kind of plug our Islands of Opportunity Alliance conference that is going live at 5 o'clock tonight. So like you guys, our face-to-face conference had to get moved to a virtual platform. And so what we've done is I've just put together a conference of student presentations that are pre-recorded. And when you go to our link, which is down at the bottom there of the flyer, you'll see all of the names of the students from all of the different Islands of Opportunity Alliance partners that are spread across the Pacific as well as within Hawaii itself. And yeah, so it's students who are affiliated with us, and yeah, there's some, I'm so proud to say that there's some really amazing presentations that go in line with what you guys, you awesome students have presented today. And thank you so much, kia ora koutou, for your work. And you know, watching you guys, it's like, it's just such a good feeling of knowing that the future of STEM is in really good hands. And yes, so thank you so much for yeah, giving us this space, Linnea, to plug our little conference.
Of course. And I've put, for those of you who are interested, I put the link in the chat so you can click on there and check it out. Again, none of what we do could be done on our own. So it's through partners and friends that we're able to do this. So mahalo. Thanks, Marata.
Bye.
So all right, folks, that is the official end of our 2020 PIPES Symposium. We're gonna take a break, but this Zoom is gonna be open and online. Folks can grab lunch and sort of come back and join us. And our interns will be here, our staff will be here. We'll figure out the logistics of what a Zoom lunchtime talk story looks like when folks get back, but feel free to jump on and ask questions however you folks like. And if there are multiple conversations going on, we can always throw folks into breakout rooms. So mahalo.